Hello and welcome to this cytokine lecture number 2. Let us first look into what we covered in the previous lecture, and in the previous lecture one of the important topics that were covered was to look at the families of the cytokine receptors. So, broadly these receptors were classified into the following families called as the immunoglobulin superfamily of receptors, class 1 cytokine family of receptors, basically otherwise known as hematopoietin receptors, which are the major class of receptors in the immune system, class 2 cytokine receptor family which binds to interferons, then you have the TNF receptor family, the toll-like receptor family, and the chemokine receptor family. So, what we will do in this lecture is to cover some aspects about the function of these receptors and how they are regulated, and then look into some of the aspects of how cellular subsets are regulated by these receptors or cytokines. So, basically just to cover a little bit of the previous lecture, we had the IL-6 receptor subfamily which shared a common subunit called as the GP130 subunit, as shown again in this slide. This particular subunit is shared between 4 different kinds of receptors which are specific for IL-6, IL-7, ciliary neurotrophic factor, and leukemia inhibitory factor or oncostatin M factor. Going on, looking at the GM-CSF receptor subfamily of the class 1 family of cytokine receptors, they have a common beta subunit. This common beta subunit is evident over here, which is the longer subunit in this particular receptor, and the alpha subunit making up the component of the GM-CSF alpha subunit. Then you have the receptors for IL-3 and IL-5 having a common beta subunit but another subunit that binds specifically to the cognate or specific cytokine in question. Now, why do these different receptors have common subunits? What is the meaning of this and what is the function of sharing of these different kinds of subunits? It so happens that many of these or all of these cytokine receptors, especially the class 1 receptors, you have different subunits. Many of these subunits bind to their cytokines specifically. For example, they have an alpha subunit or an alpha chain that can bind to its specific receptor by itself, but this binding, as you can see over here in these 3 particular cases, signifying the same 3 receptors that you see over here in the previous slide for the GM-CSF, IL-3 and IL-5. So, which have been designated here in different symbols or different objects, circle, triangle, and a kind of a trapezium. So, you have the alpha subunit which is anchored into the membrane, and this binds to its specific subunit, but this binding is one of a low affinity. In other words, it is not very tightly bound. The Kd values are less, dissociation constants are less. Once this binding occurs, there is another subunit called as the beta subunit, which is seen in the previous subunit that has a longer intracytoplasmic tail, which is meant for transducing the signaling events upon binding to its appropriate cytokine ligand. So, that is shown over here, and this is the beta subunit having the longer intracytoplasmic chain. So, there are 2 subunits. Now, these 2 subunits bind to each other. Once the cytokine binds to the alpha subunit, the beta binds to this alpha subunit, making this affinity stronger. So, a low affinity binding is converted into a high affinity binding and has the ability now to transduce the membrane signaling events that ensue due to the cytokine binding of this receptor. So, that is basically how these various cytokines function which have common subunits. So, you have a ligand binding subunit which is specific for its ligand, but it cannot transduce the signal. But upon binding to the transducing subunit, signal transducing subunit, the affinity of the receptor becomes very strong, and this affinity then results in the cascade of events that follow on the downstream events that follow beneath the membrane. We will come to that in a little while. So, what is the function or the effect of these common subunits? The effect of sharing these common subunits actually has to do with how cytokine function can be modulated. For example, here is shown in this slide, you have 3 alpha subunits, and this alpha subunit can bind to its specific ligand like IL-3, and you have 3 other alpha subunits in green, light green, that can bind to its own specific ligand, the GM-CSF ligand. So, on the membrane there are equal let us say there are equal this is a hypothetical situation in order to explain the effect or the functional consequence of such sharing of these common subunits. So, on the membrane you have equal concentrations, let us say, of these alpha subunits to 2 different lymphokines. On another location or on the cell, on a different part of the membrane, you have these beta subunits sitting there, let us say there are 3 of them. Now, at any given situation, because the entire immune function is driven by the concentrations of lymphokines that are available locally, let us say in this particular situation there is more IL-3 molecules than the GM-CSF molecules. So, you have 6 of these compared to 3 of these. So, what happens there, because of the higher concentrations of IL-3, they will bind all their subunits, because this binding is concentration dependent. However, the lower concentrations of GM-CSF will only lead to the binding of probably one of these or a fraction of the subunits that are available on the surface, the ligand binding subunits. Now, when these 3 are bound by the cytokine, the available number of signal transducing subunit or the beta subunit then binds to these alpha, but they cannot bind to the unbound alpha subunits of GM-CSF. They can only bind to those that are bound to the GM-CSF and make it a high affinity interaction. So, at a given situation when IL-3 concentrations are higher, you will find that the full functional subunit that is capable of transducing the membrane signaling events are actually the ones that are bound to IL-3, because the IL-3 concentrations are higher. So, in other words, the higher the concentration of a cytokine, it sequesters the beta subunit and therefore, makes the availability of the beta subunit lower and lower for other cytokine subunits. So, the transmembrane signaling or the downstream signaling consequences are taken over, or there is a majority of events that are transduced by this particular IL-3 cytokine. Therefore, you will see that depending upon the concentration of the lymphokine, one can see how the effects of the final functional consequences of that lymphokine will predominate. So, therefore, the cytokine actions of at any given point on any given type of cell will then depend upon all the different kinds of cytokines that are present in that local environment, and therefore, one refers to it as a network kind of function. So, this is the IL-2 receptor subfamily, which shares a common gamma subunit, which was shown to you in the last lecture. So, the IL-2, IL-15, IL-7, IL-9 and IL-4 are the members of this IL-2 receptor subfamily, and they share these blue subunits, which is the gamma subunit, and the IL-2 receptor alpha is shared, the IL-2 receptor beta is shared between IL-2 and IL-15, but the alpha subunit is the one which is binding to the ligand. In fact, the cytokine signaling via the IL-2 receptor has got a very unique property, and that is how the IL-2 receptor or the IL-2 action is modulated on a given cell. Once the availability of IL-2 is taken for granted, it is the receptors that go up and down in order to modulate the amount of IL-2, because of inflammatory situations or pro-inflammatory situations in variety of diseases, a particular lymphokine may predominate. For example, when there is a lot of IL-2, that would actually lead to hyperproliferation of many cell types, which the cell would not be able to tolerate, and therefore, in such situations as in other fields of biology, you have receptor modulation. So, if the receptor goes down, then even if there is a lot of ligand present in the local environment, they will not be able to function or give out their functional consequences via transmembrane signaling events, and that is essentially what happens with IL-2. Now, when you look at IL-2 signaling, you look at the T cell growth factor acting on T cells, you have a T cell that is normally present, which is naive, and therefore, as I told you in the last class, it is not receptive to the presence of IL-2 in the medium. But once the IL-2 receptor is available on the cell surface, then that receptor is going to bind to its IL-2 and bring about proliferation. So, once a naive T cell is activated, so you have a T cell, and if it is activated by antigen, which is the first signal. So, the antigen plus the co-stimulatory molecule via the CD28 signaling leads to proliferation and secretion of IL-2. So, that is the the preliminary event which leads to T cell proliferation, or the end result of T cell activation. So, if you were to assume that T cells, once they start to proliferate, if you go on providing exogenous IL-2, which is now available by cloning, recombinant DNA cloning methods, you can take this IL-2 or buy this IL-2 and keep on giving it to this T cell culture. And a very interesting phenomena is seen in such a situation. If you plot the number of CPM of tritiated thymidine incorporated, basically because you are looking at the proliferation of cells, which will lead to the replication of DNA, and therefore, tritiated thymidine specifically gets incorporated into this macromolecular DNA, and this is what is measured by filtration. So, you have an acid precipitable thing, or you catch them on glass fiber filters, which is then counted in the counter. So, you get increasing CPM incorporated into the DNA, which is a measure of the proliferation of that population of T cells. So, if you look at time after antigen activation, provided of course, you have a lot of IL-2 added to the culture, you will see that the CPM incorporation will go up in the beginning stages, but after a while it will plateau and subsequently it comes down. So, therefore, if you take antigen activated cells and you provide them with a continuous supply of IL-2, it does not result in continuous proliferation over a period of time, which does not happen. However, it goes up in the beginning stages and then it comes down. What is the reason for this? The reason for this is that although IL-2 is there, initially more of IL-2 is bound and brings about the signaling events, which leads to activation of several types of genes, including IL-2 gene induction, which leads to the secretion of more of IL-2, therefore, more IL-2 accumulates in the medium. However, after a while, the IL-2, the very IL-2 receptor that binds to this IL-2 starts to modulate its function. In other words, it cannot bind as much of IL-2, or it cannot lead same strength of functional consequences that was happening in the initial stage after antigen activation. This curve can of course be restored by again providing with antigen. So, if you provide again with antigen to the antigen presenting cell, again you will see that the incorporation goes up. What is the reason and what is the mechanism for such a phenomena? So, you will see that in most cells, for example, in T cells, you will have there are 3 subunits to this IL-2 receptor, which is called, as you saw in the previous transparency, it is called as the alpha subunit. This alpha subunit is also known as the T cell activation antigen, TAC antigen. The reason being that this alpha subunit is what predominates on activated T cell populations, and initially when they immunize these T cells, in order to identify the structure on the cell surface that would actually bind to the IL-2, they found that all the monoclonal antibodies that were derived from such an immunization with activated T cells actually bound to an antigen, which is now characterized as the alpha subunit of the IL-2 receptor, and therefore, because they predominated on activated T cells, this was called as the T cell activation antigen or the TAC antigen for short. So, when you refer to these, they call it as the TAC antigen or the anti-TAC antibody. But surprisingly, actually this alpha subunit is not the one that is responsible for the consequences of T cell proliferation or the downstream events. It so happens that the IL-2 transducing subunits are actually other subunits called as the beta and gamma subunits. So, this beta and gamma subunits are the ones that actually transduce the signal, as evident in this drawing by the longer intracytoplasmic tail. So, what happens is that on a normal naive T cell, you do not have as much of the alpha subunit, but it has the beta subunit and the gamma subunit at very low concentrations. So, what happens is that this particular beta and gamma subunits are able to bind to the IL-2 ligand with what you call as an intermediate affinity. So, this particular subunit combination is of intermediate affinity. Having a Kd value of approximately 10 to the minus 9 molar. So, you will see that when you look at this intermediate affinity, the IL-2 binds to its receptor, and after this binding is over, the alpha subunit starts to get induced on the cell surface. In other words, there is a transmembrane transducing event, which then induces this particular gene on the cell surface, and more of alpha subunit comes out on the cell surface, which binds to this beta gamma subunit, making this particular complex a high affinity receptor, which has something like 10 to the 11 molar Kd. So, you will see that from the low affinity, you have the conversion by the binding of this TAC antigen converted to the high affinity receptor on the cell surface. This alpha subunit has got a short intracytoplasmic segment, and therefore, is the low affinity subunit. So, what happens is that as the cell gets activated and starts to proliferate, make more IL-2, as time goes by, the concentration of this TAC antigen starts to increase on the cell surface. When it increases on the cell surface, it starts to bind more and more, or so to say scavenge the IL-2 that is there in the medium. but once it binds to more and more of IL-2, it is unable to transduce the signal, and therefore, the cell becomes non-responsive, although IL-2 is being bound by the subunit. So, this gamma subunit is actually the one that is present to a certain extent constitutively in the beginning, but is missing in SCID patients, severe combined immunodeficient patients, which is an X-linked disorder. They have an abnormal thymus with abnormal T cell numbers in the thymus, and they are prone to a lot of infections. This gamma subunit is also very low in Chagas disease, which is caused by Trypanosoma cruzi. In fact, they have found by infecting in vitro with Trypanosoma cruzi, they have found that this subunit, the gamma subunit starts to, the IL-2 receptor starts to decrease on the cell surface in such situations, and therefore, there is no IL-2 mediated signaling, and that leads to lack of T cell proliferation, and therefore, problems with T cell responses. So, this is basically how a T cell response is actually regulated, and is of course, responsible for the modulating effect on the proliferation that I just explained in the previous slide. So, going on further, you see that apart from this sort of signaling events that are regulated by the availability of different kinds of subunits on the cell surface, there are other places where there could be regulation, and that is the transmembrane signaling event itself. There are several events that can be regulated, and to look at the cytokine or the class 1 cytokine receptor family in general, as well as the interferon family in general, you find that these kind of receptors are actually going through what are called as Janus activated kinases as well as STATs, or signal transducer and activator of transcription. So, this is called as a JAK-STAT pathway. So, what happens generally in these receptors is that the alpha subunit first binds the ligand. This binding of the alpha of the ligand by the alpha subunit actually changes this alpha subunit in such a way that it dimerizes with another subunit. So, the alpha and beta subunits come together, and this coming together actually binds the ligand more tightly, which is what we saw in the previous slide, where we looked at how the alpha and beta subunit come together to form the high affinity binding receptor. So, this dimerization of the receptor follows this ligand binding, and this dimerization then leads to downstream consequences beneath the membrane, which are basically phosphorylation events mediated by kinases. So, these kinases are nothing but the Janus activated kinases. This is a situation or a step where tyrosines that are present in the receptor of the cytokine, in this case the gamma interferon receptor, is phosphorylated at a multiple tyrosine residues. In the whole event, of course, the kinase itself autophosphorylates. This phosphorylation then leads to another step, which is characterized by the docking of this particular protein called as the STATs. We will see that in the next figure. This docking of the STAT to the receptor brings it in close proximity to the tyrosine kinase, or the kinase activity of the Janus activated kinase, which starts to phosphorylate the STATs. Once the STATs are phosphorylated, they are now available to dissociate and bind to themselves, causing a dimerization of the STAT protein itself, STAT subunits. This dimerization then allows these dimers to get into the translocate into the nucleus, and subsequently it binds to the upstream promoter elements of various promoters that are promoter elements that are present in upstream of various genes that are activated by these cytokines. So, looking at this in a little graphically, you find that in the case of interferon, as you can see here, this is the alpha subunit and this is the beta subunit. So, initially these are not bound by interferon. So, one of these subunits starts to bind interferon, and this binding of interferon by the subunits starts to allow this dimerization to occur. In other words, the alpha and beta come together with binding of interferon. This binding of interferon, as I told you in the previous slide, activates this what you call as Janus activated kinase, which you see as a button there. So, you have the receptor over here. This is the Janus activated kinase. So, this binding event activates this kinase. The function of a kinase is phosphorylation. So, this once the enzyme is activated, it starts to phosphorylate. What does it phosphorylate? It phosphorylates the receptor subunit and it phosphorylates itself. So, there is autophosphorylation as well as phosphorylation of these receptor subunits. Now, these are the STATs which I told you in the last slide. These have got in them SH2 domains, or these are called as SH2 proteins. These have the ability to bind to these phosphorylated tyrosine residues, which is shown over here. So, therefore, these subunits come and bind to these subunits. In the whole process, they are brought in proximity. You see, the membrane is such a large space of the cell. So, all these phosphorylation events are actually mediating the movement of the STAT towards these tyrosine residues. So, they have become proximal with the tyrosine kinase. Once there is proximity, the STATs gets phosphorylated on the tyrosine residues, and this is what is seen over here. And these phosphorylated STAT subunits now start to come together. They come out from there and they come together to dimerize. This dimerization event unlocks certain sequences which allow them to go into the nucleus, this being the nuclear membrane. Once they go inside, they start to bind to various kinds of DNA promoter elements which are upstream, enabling the transcription of that gene. So, this is how the general cascade of events occurs for these cytokine receptors, especially for the class 1 and class 2 family. Of course, when you look at the TNF family of receptors, they have their own receptors. Basically, these are bringing about death, and therefore, downstream signaling events have subunits which are composing of what are called as DDs or death domains, because these are proteins that mediate the final function of apoptosis. So, you have the details of this cascade need not be covered in this class, because this is not a signaling class, but basically leading to apoptosis. That signaling pathway is different from that what you see for interferon and IL-2. Basically, the end result of the TNF pathway is the activation of the transcriptional factor NF kappa B, which gets into the nucleus, translocates into the nucleus again after being phosphorylated, and the removal or disengaging from the I kappa B subunits, which are degraded by the ubiquitin pathway. So, this then translocates to the nucleus, just like what happens with the STATs, and the activated NF kappa B binds to its cognate elements on the NF kappa B inducible genes. So, if you were to look at what are the different kinds of Janus activated kinases or STATs that are involved for the different kinds of cytokines, you find the list given over here. For IL-2, it is JAK1 and JAK3, which are associated with the receptor subunits, and downstream events include the STAT5. IL-4, which basically IL-2 and IL-4 have many overlapping functions because they share their subunits. It has JAK1 and JAK3, but STAT6 is the one that performs the downstream signaling and translocation into the nucleus. IL-6 uses JAK1, in some situations it uses other, but basically it uses JAK1 along with STAT3. IL-10 on the other hand, JAK1 and another tyrosine kinase called as TYK2, which is coordinated downstream by STAT3. IL-12, another very important cytokine, IL-10 being characterized as a kind of an immunosuppressive cytokine, but IL-12 being a pro-inflammatory cytokine, IL-10 being an anti-inflammatory cytokine. IL-12 activates JAK2 and TYK2 along with STAT4 downstream signaling event. IL-3 utilizes JAK2 along with STAT5. Gamma interferon utilizes JAK1 and JAK2 along with STAT1. IFN alpha beta utilizes JAK1, TYK2 along with STAT2. So, these are the different kinds of cytokine receptor associated proteins that you find with the with the different cytokines that are listed over here, and the pathway that was described in the last couple of slides. So, going on further, a very important aspect of T cell immune responses and cytokines is one of classification of different kinds of T helper cells. So, once you look at the CD4 positive cell, we call them as a T helper cell, but you see there is so much of heterogeneity even within a certain kind of cell. So, one can classify pupils or students belonging to a particular class. Let us say they are studying in the 5th standard, but then when you go and look at the pupils themselves, you can even classify them further as very hard working and being in the first 10% of the class, as to others who do not study or are not able to pass the exam. So, there is heterogeneity even within the class of pupils. Similarly, here with this T cell populations you have heterogeneity, and they were actually initially characterized by Mossman, who classified or who observed that T cells actually secrete different kinds of cytokines. Based upon the secretion pattern of these cytokines, one can classify or separate or purify T cells which are of different types. And it so happens that in this particular slide you have two different kinds of T helper cells called as Th1 and Th2. These two different kinds of T helpers have a very important bearing and consequence of on the immune system. So, if you look at Th2 and you see the red marks or the red ticks over here, you will see that these are the cytokines that are secreted by Th2, IL-4, IL-5, IL-10 and IL-13. On the other hand, these cytokines are not produced or secreted by the other type of T helper cells. In other words, there is a dichotomy in the type of cytokines that are secreted by these two T helper subsets. So, if you look at these cytokines IL-3, GM-CSF, gamma interferon and TNF beta, you will find that these are secreted by the Th1 subset. So, you also have TNF alpha or the TNF also being secreted in such situations of inflammatory situations. So, you will see that these are peculiar to the Th1 subset, whereas these are peculiar to the Th2 subset. Now, as we went into the functions of all of these lymphokines, you will find that all of these would help B cells, whereas gamma interferon would help in IgG blocking of switching to IgE, and therefore, it would block these when there is a abounding of these cytokines or T helper 1 predominance, it would not allow the B cells to switch to IgE responses. So, these are some of the situations where you see that macrophages are activated by Th1 cells because of the presence of gamma interferon. Cytotoxic T cells are activated by Th1 response. Remember, cytotoxic T cells are actually CD8 positive, but these Th1 and Th2 are CD4 positive cells. Now, these cytotoxic T cells, or for that matter CD8 bearing cells, also have cytokine secretion patterns, but in a major way these are the ones that have been defined very clearly for the CD4 subsets. So, the DTH or delayed type hypersensitivity reactions are actually facilitated by a Th1 response. In a Th2 response, of course, you have more of IgE because you have IL-4 and IL-5, which would help in the switching differentiation of B cells, class switching of B cells, that would give IgE. So, therefore, total antibody production is facilitated by Th2 cells. So, you see there are two different kinds of Th2, and this having a very important bearing on the outcome of immune responses after antigen activation. So, what is the nature of this particular regulation of these T helper subsets? So, you will see that you have what you call as a naive Th0 cell, which normally would secrete IL-2, IL-4 and gamma interferon, and these are actually differentiated to form effector T helper 1 or so called effector T helper 2 cells, which actually secrete the final lymphokines they have to secrete. Now, there are two types of dendritic cells which are basically antigen presenting cells, which drive the development of the Th0 cells or the precursor T cells to finally the effector T helper cells. So, you have these Th0 cells which develop to become the effector Th1 cells in the presence of IL-12. So, more the IL-12 in the medium, there is a differentiation or development of the effector Th1 cell. Now, for this IL-12 to act, you need the transcriptional factor called as Tbet and the participation of the STAT4 molecule. This is facilitated by antigen presenting cells which are myeloid like, and they are called as DC1, as opposed to DC2 which are lymphoid like, but they influence or help in the differentiation of this Th0 to form effector Th2 cell types. Now, this is facilitated in the presence of IL-4. So, if IL-4 predominates in the medium, then you will see the differentiation of this Th0 to become effector Th2 cells. Now, the Th2 cell differs from the Th1 cell, as we saw in the last slide, in that they secrete IL-4, IL-6 and IL-10 as predominant cytokines, and the Th1 secreting IL-2, gamma interferon and TNF as the predominant cytokines. So, these lymphokines actually lead to predominantly inflammation, and these actually are called as anti-inflammatory because they further total antibody responses or humoral antibody synthesis. So, how is this pathway regulated? This STAT6 and GATA3 are the ones. GATA3 is a transcriptional factor. So, when GATA3 is active, then you have Th2 formation. When Tbet is active, you have Th1 formation, and STAT6 is predominantly active for this pathway of Th2 effector cell differentiation. So, if you look at this, it so happens that the lymphokines that are responsible for this kind of differentiation into Th1 and Th2 cells are actually cross inhibiting the two pathways. In other words, they are mutually trying to inhibit each other. So, this IL-4, which was responsible for the differentiation of effector Th2 cells, you find that the same IL-4 is able to inhibit the differentiation of Th1 cells. So, if there is more of IL-4 in the medium, you have a block of differentiation of Th0 into Th1. Basically, you find that there is more and more of IL-4, more and more of IL-6 and more of IL-10. So, more the IL-4, you get more of Th2 activity and an inhibition of the Th1 type. On the other hand, if you look at the Th1 effector cytokine, basically gamma interferon, which it secretes, there is more of gamma interferon that is synthesized. It has a block on the development of Th2 cells. On the other hand, more gamma interferon actually accelerates or facilitates the differentiation into Th1 cells. So, more Th1, more gamma interferon, and therefore, the pathway is driven basically towards a Th1 bias. Now, all these events are actually dictating the outcome of disease. We will come to that in the next few slides. So, going on further, you find that the same gamma interferon also has a blocking on the GATA3 transcriptional factor. In fact, that block is what blocks this differentiation pathway. On the other hand, this enhances the IL-12 mediated T-bet activity. So, if you go on further again to show you that IL-4 does the job by inhibiting T-bet transcriptional factor. So, these are the ways by which these two cell types are actually mutually inhibiting each other. So, going on further, in addition to Th1 as well as Th2 cells, there is a very important cell type called as the Th17 cells. Now, these Th17 cells are very important and they have found it to predominate in actually antibacterial immune responses. They are induced to develop in the presence of a cytokine called as IL-23. Now, both IL-4 as well as gamma interferon can inhibit this differentiation. So, if you look at Th17 cells, after they differentiate, they secrete copious amounts of IL-17. Now, these cells are CD4 positive and FOXP3 negative. FOXP3 again is a transcriptional factor that is present on a different cell type. We will come to that in in the next slide. So, you see IL-17 induces the secretion of pro-inflammatory cytokines from fibroblasts, epithelial cells as well as endothelial cells, for example, IL-6. So, basically if you were to look at the differentiation of IL-17 or Th17 cells, you look at the differentiation of these different kinds of T helper cells, Th1, Th2 as well as IL-17, you see that basically a cell, the Th0 is having, or the precursor will be having a receptor for IL-12 receptor beta 1. Once this differentiates, gets activated by antigen, it puts out beta 2 receptor, which is for the IL-12 cytokine. Once this happens, this particular cell is able to bind IL-12. So, if it is able to bind IL-12, there are different ways by which it can go for differentiation. If IL-12 predominates over there, it differentiates into Th1. If in the milieu you have IL-4, this differentiates into Th2. On the other hand, if IL-23 predominates in this situation, then they develop into Th17 cells. So, this is how you have the development of the precursors which are not yet become effector cells, which then differentiate into Th1, Th17 or Th2 based upon the predominant presence of IL-12, IL-23 or IL-4. Of course, the presence of these factors are associated with the induction of the appropriate type of transcriptional factor, whether it is T-bet or GATA3. So, going on further, the overexpression of IL-17 is actually observed in rheumatoid arthritis, systemic lupus erythematosus and asthma. So, there is a small, so this has to be rheumatoid arthritis, and then of course, you go on further to look at a different kind of T helper T subset, which is now you had three subsets of T cells called as the Th1, the Th2, the Th17, now you have the Treg cells. These are regulatory T cells, which are basically meant as a regulator of immune responses. So, if you have an overreaction or a hyper immune response, especially in terms of inflammation, you have these Treg cells that play a very important role in trying to suppress an ongoing immune response. So, they are important for regulating immune responses and hyperimmune stimulation states. So, these Treg cells again come in different subsets having different surface markers, but basically they are CD4 positive and FOXP3 positive, a transcriptional factor that is characteristic of these Treg cells. Some express the CD25 antigen or the TAC antigen. What is the consequence of expression of this TAC antigen? The consequence of the presence of the TAC antigen on the surface of these cells is that it binds the IL-2 and therefore, it scavenges the IL-2 and makes it unavailable for the neighboring T cells that are actively proliferating, and therefore, the inflammatory situation where proliferation is occurring is actually comes down, because more of IL-2 is sequestered onto this subunit, and basically these cells will not proliferate because this TAC antigen cannot mediate the transmembrane signaling events, and therefore, no gene induction. So, the Treg cells are induced to develop in the presence of TGF beta and control effector T cell functions like what I told you here, in addition to other kinds of mechanisms of function. So, therefore, there are 4 major types of T subsets called as the Th1, the Th2, the Treg cells and the Th17 cells. So, going on further, there is a bias. How is this Th1 and Th2 play a role in disease mediation or disease states? So, you find that in various disease as well as in clinical situation, one finds a predominant bias towards one cell type or the other. So, you find certain diseases actually has a Th1 bias, and certain other diseases has actually a Th2 bias, and certain other stimuli actually promote the development of Treg cells. So, all this is actually mediated at the level. There are several steps that is thought to determine this sort of bias, depending upon how the antigen binds in terms of its affinities and how strongly it binds to the T cell receptor. And you find that in these situations, examples being in Leishmania, especially in B6 mice, you find that Leishmania infection actually does not kill the mouse, but they are protected or they do not come down with death. On the other hand, another strain of mouse called the BALB/c mice, when infected with Leishmania, they are susceptible to this organism and lead to death. So, when you look at these mice, you find that their lymphoid cells or T cells are actually having basically a bias towards Th2 type, but the healing mice, which are the B6 mice, have a bias towards Th1 type. As I told you earlier, these two cell types are cross inhibitory. So, IL-4 inhibits the Th2 type and gamma interferon actually inhibits gamma interferon inhibits the Th2 type, I am sorry, and IL-4 inhibits the Th1 type. So, you can actually give anti IL-4 to BALB/c mice. So, if you inject to BALB/c mouse, now this BALB/c mice is now having a Th2 bias. So, there is a lot of IL-4 that is influencing or encouraging the formation or differentiation of Th2 cells. So, if we block this high amount of IL-4 by injecting anti IL-4 antibodies, the IL-4 is neutralized, and therefore, there is a block in Th2 development and it gets converted to Th1 because of the presence of IL-12 in that local medium. Remember that all these different situations are of relative degree. So, you find in the local environment, it is not as if that IL-12 is not present, but the concentration of IL-4 predominates over that of IL-12. So, it is a question of relative concentrations of these various kinds of cytokines that are predominating in that particular inflammatory or non-inflammatory situation. So, once this becomes Th1 type, you find that the bulb C mouse is now protected. So, you have protection ensuing because you injected IL-4. On the other hand, the opposite situation, where if you inject anti-interferon gamma in C57 black 6, instead of healing they become susceptible. Why? Because anti-gamma interferon neutralizes the interferon that is there, that is influencing the formation of Th1 cells and allows the IL-4 cells to develop. So, the other situation that has been found in humans is leprosy is of two types in humans, called as tuberculoid leprosy and lepromatous leprosy. Usually tuberculoid leprosy leads to a kind of a healing situation where the number of organisms actually decrease, whereas in lepromatous leprosy there is much more nerve damage because of the organism, and it leads to more deleterious consequences. And it so happens when one does a northern blot or looks at the concentration of mRNA that is there in these infected tissues from these patients, you find that the tuberculoid leprosy patients predominate in a Th1 response, which is leading to a healing type of response. And a lepromatous leprosy patient has a predominant bias for Th2 response, which leads to a kind of susceptibility. So, you see that many of these bias is actually associated with the outcome of diseases, protection or susceptibility. But it is not to say that always the Th1 responses are always going to be beneficial. In fact, there are many situations where Th1 response is not beneficial, where too much of Th1 response actually leads to a lot of inflammation. So, it becomes pro-inflammatory, causing a lot of inflammatory responses and leads to necrosis or deleterious consequences on the tissues. Inflammation is what causes it. So, a more of pro-inflammatory cytokines leads to damage to the tissues and is not preferable. So, an immune response is actually a balance between inflammatory responses and the other type of responses. So, it is a balance between Th1 and Th2, and different disease situations would receive a better prognostic consequence or a better prognostic effect if one looks at what type of Th bias is required for protection. So, if one can convert a bias which is deleterious to a bias which is going to be advantageous, would be very preferable in the clinical situation, and in fact, that is what is being tried in the clinical situation involving cytokine therapies. Some of these clinical examples are, for example, you have gamma interferon being tried, the name being given to this sort of drugs is called as Actimune, which has been tried in chronic granulomatous disease, where you have a problem with the antigen presenting cells and the macrophages, and you have interferon beta, which is called as beta seron or A1X, which has been tried in multiple sclerosis. Interferon alpha, which has been which has been tried in antiviral therapy for chronic hepatitis B and C as well as in hairy cell leukemia, and the products are called as Rofiran, Alferon or Intron. Then you have Neupogen, which is nothing but granulocyte colony stimulating factor as a supportive treatment for bone marrow transplantation. Then you have GM-CSF, then you have chimeric TNF receptor TNF receptor, which is as a fusion with the IgG constant regions. This called as Enbrel, has been used in the case of rheumatoid arthritis. Monoclonal anti-TNF alpha receptor has been used in the case of rheumatoid arthritis and Crohn's disease. Of course, needless to say, many of these cytokines are toxic when given systemically, and therefore, one has to be careful in how it is administered and in what concentration and in what mode. So, summarizing this class, of course, to go into the various kinds of clinical situations, it would be of more detail and therefore, not able to cover in a short space of time. We have covered in these 2 lectures of cytokines, we have covered the nature of cytokines, their properties, and described some of the salient properties of some of these major cytokines, and then looked at how these cytokines are classified and what sort of receptors they bind and what are the classes of these receptors, and looked into how these receptors act and what is the meaning of how they share their subunits, and then went on to look at how these different cytokines can actually classify different kinds of T helper cells, and how the T helper cell bias is actually observed in different kinds of disease situations, and how they can be altered by using the cytokines themselves to alter the differentiation of these various T subsets. Thank you very much.